So to get started, I thought it'd be useful to explain exactly what I'm talking about here. So there's a new glossary around terms for fertility, infertility and its treatments. And that's really helped us to understand in research what we're all talking about. So assisted reproductive technologies are any intervention that involves in vitro handling of both the human oocyte and sperm or of embryos for the purpose of reproduction. At least 95% is in vitro fertilization with or without intracytoplasmic sperm injection. So I do occasionally slip into using IVF rather than ART, but most of what we're talking about is IVF. So about 1 in 6 couples worldwide are infertile, and since its first implementation, the use of ART has increased and is now considered a routine effective treatment for infertility. Around 10 million people worldwide have been conceived by ART. In Europe, about 1 million live births per year have been conceived by ART, with variation across different European countries. So up to 8 children in Europe, sort of between 1 and 8 children in Europe. If you go to a primary school class, that's usually around 30 children will have been conceived through ART. In the UK, it's on average 2 per class, for example. The graph here is showing that it is still through the 1990s and up to 2018. The drop off at 19 is just with the early months of 19, and the data are not available beyond that. We continue to see this increase in usage, particularly in women under the age of sort of early mid-40s. I wanted to show some equivalent type of data for the US, and this is taken from a paper we did looking at the impact of financial crises on the provision of ART, and with an idea of predicting that in relation to COVID and the current financial crisis. But for the purposes of this talk, you can ignore the arrows about the financial crisis. It's really showing a similar pattern in the USA to what we see in the UK and other countries, that there is still this increase in use of ART and therefore increase in the number and the proportion of live births that are conceived from ART, against a background for this decade in the US of a pretty constant just under 4 million births per year. So as the use of IVF ART has become more common and the proportion of couples treated who have a live birth increased, there's been a sort of shift towards more research and interest from couples and clinicians, people who provide ART, of interest in what might the health consequences be for the mother and for the offspring in having, in the mother's case, been treated with ART, in the offspring have been conceived by ART, both in terms of perinatal health and also future health of different natures. But what I'm going to talk about here is cardiometabolic health. As with any of our epidemiological analyses, this research is potentially biased by confounding selection bias and misclassification. And in addition here, when we start to become interested in cardiovascular, cardiometabolic health or mental health, other outcomes going into adulthood, there are still, by and large, too few people who have been conceived by ART to have those sort of later life diseases. So most of the research in this area comes from 2 types of study, either large-scale record linkage studies, or what I've called here clinical cohorts. And what I've tried to summarize here is the somewhat different nature of the types of biases in these 2 studies, which for me makes it interesting, because then we can compare results from these different types of study with their different biases. And if we get consistent results, we have more confidence in those results. But just to summarize, in terms of compounding, often the electronic linkage studies will have some unobserved or maybe not well measured compounding. So high risk of residual compounding. For example, they may not have detailed information on smoking or body mass index or other types of behaviours. In the clinical cohorts in relation to compounding, these are cohorts where the couple undergoing ART have been recruited and then followed forward, including following their children should there be a successful live birth. And in analyses then that start to answer these questions about differences between an ART conceived and a naturally conceived child, the comparison group of naturally conceived children are often from friends and family. And even if they are a more representative group of the children who would have been selected had they been conceived by ART, they're often recruited at that later age when people are interested in looking at risk factors such as blood pressure in childhood. And it means that you're less likely to have any preconceptual data that you would have in detail in the ART group. And you may also have much less information around the time of pregnancy and perinatal health. So a different type of compounding, but for both worries about residual compounding. In the electronic record linkage studies, we can do these within SibShip analysis to try and control for family level compounding, which I'll talk quite a bit more about in the later slides. So in terms of selection bias for the conventional, what I call population analysis, the multivariable regression between unrelated individuals, I think selection bias is not likely to be a problem when you have these whole population linked data. But, and I'll come back again to this, I think there's potential for a particular type of selection bias within SibShip analyses. I've mentioned the nature of the comparison group in the clinical cohorts, and that, as well as introducing some compounding, can introduce some selection bias. By contrast, the clinical cohorts are likely to have really good detailed information about the whole type of ART, and wouldn't be confusing ART with medically assisted reproduction that might just involve having treatment to stimulate the ovaries, which can be a problem in the electronic record linkage studies. And then just to add a bit more complexities, and I quite like doing research where there's something that has to make you think about all these different things. There's a variation over time in treatments and compounding factors that I think means we have to be cautious when we are comparing different types of study and different populations. So, for example, socioeconomic position, conceivably a confounding factor, but it may be strongly related to the ability to pay for ART, and that varies massively between different populations. In the UK, where I work, we have what's called a postcode lottery, because although the national guidelines say that up to 3 cycles of ART should be provided by the National Health Service, this is an area where what are called local commissioning groups can override that, and they do. So you can live on opposite sides of the same road, and on one side there may be National Health Service provision of 3 cycles, on the other side no cycles. And that varies hugely between countries also. Smoking and body mass index, I would naturally, I think most people would think of as a confounder that increases, if you've got higher body mass index and smoking, your risk of being infertile and therefore having ART, and will increase your risk of some of the adverse pregnancy and perinatal outcomes and future cardiometabolic health. But there's also variation in provision of ART by these risk factors for infertility and for success of ART. So in the UK, whether self-funded or National Health Service funded, you have to be a cotinine Prova non-smoker for the last 3 months before treatment, and have BMI in a healthy range. So then that sort of changes the whole notion of these being confounders. And then lastly, one of the big issues around ART now is what are called add-on treatments. Lots of different things that can be added to the standard IVF with big promotion of how much they're going to improve the live birth rate, but really no evidence. And that's continuously changing, and an area again where we need good research, but it's difficult to research. So we can't do randomized control trials comparing any type of ART versus natural conception. In theory, we could do randomized control trials of different types of ART, and there is currently great interest in looking at the effect on live birth rates of intracytoplasmic sperm injection in couples where there's no evidence of male infertility on live birth rate. In the UK, it's really taken off for everybody. In the Nordic countries, it's used only in the context of male infertility. It's more expensive, whether it's the National Health Service or the couple paying for it, and it's not clear whether it makes a difference. But in practice, these trials are really hard to do. So Ben Moll and colleagues did a review of all of the randomized control trials in this field and showed that around 1% of the trials were powered to be able to detect the key outcome that couples want to know, which is live birth rate. And to do that, you really need large studies, because despite its increase in effectiveness, it is still only between 20 and 40% of a treatment cycle that results in a live birth rate, determined by prognostic factors, in particular the woman's age and whether donor oocytes are used. And then there are sort of interesting issues, because this is quite a loaded area. I think you're quite vulnerable when you want a child and can't have one, and you kind of somehow given strong reasons to believe certain treatments are better than the other. So there were two large randomized control trials, one in Australia and one in the UK, multi-centre, that aimed to tackle, again, what was a really pressing question at the time because of concerns about transfer of two embryos, increasing the risk of twin pregnancies, and then the increased risk of small for gestational age and preterm birth in twin pregnancies. So there was a need for trials to compare a single embryo fresh transfer followed by a subsequent frozen transfer to a double embryo fresh transfer. These two trials started, and as you can see from the numbers there, they were unable to recruit anything like the numbers that they had set out. And this is recruitment. And I was really fascinated by this and did a lot of freedom of information things and got to the bottom. And basically, in Australia, the clinicians were very convinced about the single embryo transfer and had persuaded couples going for it. There'd been a lot of publicity about how important this was, and that meant that couples in Australia didn't want to be randomized. They wanted the single embryo transfer, whereas in the UK the attitude was, well, yes, as obstetricians we worry about preterm birth, small for gestational age, but in reality the vast majority of infants born preterm or small for gestational age go home healthy and fine within a day or two of being delivered. And for infertile couples, twins too may be a nice way of completing their family. So there are these real difficult issues around being able to do randomized control trials, even for different types of ART. So in my mind, we need to try and be smart about how we get good causal inference or the best causal inference we get. So I'm not particularly a royalist, but this family tree of the UK royal family gives at one extreme the idea behind these within sibling group analyses. So if we consider, for example, down here, William and Kate's 3 children, they are born into this family with privilege and high socioeconomic position and aristocracy across decades. And their mother didn't smoke in any of her pregnancies. She has a healthy BMI. So lots of the things that we think may be confounders in this field within these sibling groups are adjusted for whether you've observed them or not, because they're consistent within the sibling groups. And it works also at the other extreme of socioeconomic position. But there is no free lunch with any of our methods, and you can have problems with what's called crossover or contamination. So if either the intervention or the outcome in the earlier SIB influences the intervention or outcome in the later SIB, then that can bias the results. The nice thing about these population studies is, by and large, they show both the conventional unrelated multivariable analysis results, where we might worry about not fully adjusting for confounding, and the within sibling analysis, where there is potential control for the main confounders, the within family confounders, but they come with this other sort of bias, and it's nice to be able to compare the two. So this is an example of where we did this. This is some work I've done with the Conatus collaboration, which is this big linkage of 3 Nordic country data, Denmark, Norway, and Sweden. So you get very large numbers. This was Kirsty is a PhD student, now submitted and about to have her Viva and doing some clinical work again. And it was a real joy to be part of her supervisory team. And what this first set of results is showing is asking the question, what's the effect of a fresh embryo transfer compared to natural conception, and a frozen embryo transfer compared to natural conception on 3 perinatal outcomes: small for gestational age, large for gestational age, and preterm birth. And what Kirsty did here was compare the sibling analysis, the conventional population analysis, in a sample that we described as minimizing selection bias, because that included everybody, and a sample that we described as the minimizing observed confounding bias, because in these studies, as sort of reflecting what has happened over time in record linkages around ART, but also around pregnancy and perinatal health, is that in the more recent years, there's consistent information on body mass index and smoking in these linked data, whereas in the earlier years they weren't. So if I just focus on one set of results here in this graph, which is looking at the fresh embryo transfer with compared to natural conception, and these initial results at the top here, the math isn't working now, but look at the top red and orange results, are all looking at small for gestational age as the outcome. The very top one is the minimizing selection bias, conventional population analysis. The next one down, minimizing selection bias within SIBSHIP analyses. And then the same 2 again, but within the sample that is somewhat smaller, but able to adjust for additional confounding by smoking and body mass index. And you can see there, in relation to small for gestational age, a consistent result that being conceived by ART with a fresh embryo transfer compared to natural conception increases the risk of small for gestational age. It's quite consistent across the results. As we would expect, the sibling analysis is less precise than the population analysis, and that's because the sibling analysis is restricted to those with discordant exposure, and when you've got a binary outcome, as here, discordant outcome also. For the fresh embryo transfer, there doesn't seem to be much impact on large for gestational age. And then for the preterm birth, there's an increased risk, but in comparison to the population results, the within sibling analysis attenuate that risk for preterm birth, suggesting that with preterm birth, there may be an increased risk, but some of what's seen observationally in the conventional analysis is due to confounding. And for the frozen embryo transfer, for preterm birth, we see very similar results to fresh embryo transfer. Interestingly here, and there's more evidence building on this now, we see the opposite to what we see with fresh embryo transfer, in that the frozen embryo transfer is related to increased risk of large for gestational age and sort of null or possibly a decreased risk of small for gestational age. So then what Christy looked at in her next paper was the relationship again of fresh and frozen embryo transfer, both compared to natural conception, this time with perinatal death, and looking here on this graph sort of separately with stillbirth and then neonatal death. And in a nutshell, what the different analyses are adjusting sequentially for different things. What they suggest is that both fresh and or frozen embryo transfer ART compared to natural conception increases the risk of neonatal death, but doesn't clearly have an impact on stillbirth. And I guess the question now is, so why in that paper didn't we do the sibling analysis? And it was because, as PhD students always do, Kirsty had done a good review of the literature, and there had been 3 previous studies that had looked at any ART versus natural conception with perinatal mortality, stillbirth and neonatal mortality, and had found in the population analysis, the conventional analysis, ART increased the risk of perinatal mortality, again, similar to what we had seen, particularly for neonatal mortality. But this strange, strong protective effect of ART in the within sibling analysis. And it didn't seem to make sense. And there was various discussions about why that might be, but it hadn't been looked at in depth. And probably there wasn't the sample size to do what we then, well, Kirsty went on to do in the Conatus cohort. And the hypothesis we had was that this could be a bias due to a combination of selective fertility and this crossover or contamination. And what we mean by selective fertility is there's been lots of studies that's sort of well established that irrespective of mode of conception, when a couple experiences perinatal mortality, either stillbirth or neonatal death, within a given time frame, they are more likely to get pregnant again than those who have not experienced perinatal mortality. And crossover, I've explained to you. So it felt implausible, given ours and other studies had shown these relationships using within sibling and population analysis, getting consistent results, that ART likely increased the risk of preterm birth, and with fresh embryo of small for gestational age, frozen embryo of large for gestational age, these would be risk factors for perinatal mortality. And it just biologically didn't feel right. It didn't feel right to see such different results. So we went through a series of questions. In the biggest study in Conatus, do we get the same disparity in results? And then can we unpick what we think are these sources of bias? So do we see evidence of carryover? Do we see evidence of selective fertility? Do we see the two of them working together? And then we had an ambition of seeing if we could correct for it and show that it really was due to this bias. So do we see similar results? Yes, we do, and they're quite dramatic. So in the population analysis, it's suggestive of conception by ART increasing the odds of perinatal mortality by 26%. And that contrasts with what looks like a protective effect of 64%, which does 64% reduction, which doesn't make any sense. And you can see that if the first conception was by ART, then the rate of perinatal mortality is the same in both population, in the population of the sibling analysis. Whereas if the first conception, and these are discordant pairs, if the first conception is natural conception, then in the sibling analysis, sorry, I got it the wrong way around. This is showing the carryover. I'll start again. So if the first conception is ART, then either in, no, I'm right in the first time. Oh my goodness, sorry, yes. But basically what it shows is if your first child was born by natural conception, in the population analysis, the rate of perinatal mortality is 0.5%, slightly less than the ART. But in the sibling analysis, where the second one is then born by ART, you have this increase, this different risk. We showed this sort of carryover effect. And what you see here is that whatever the combination, the second pregnancy is less likely to have perinatal mortality than the first pregnancy. But when the first pregnancy has had perinatal mortality, but what you see is when the combination is a natural conception followed by ART, then you go really markedly from a 2.4% risk of perinatal mortality in the natural conception to it going right down to about 0.6% and similar to the other ones. So, and these are sort of statistically different across the different groups. So suggesting both that we have the selective fertility, but there's also potentially some carryover that's affecting them with perinatal mortality. This shows in particular how we see for both where the first delivery survives compared to when there's perinatal mortality in the first delivery, you have an increased risk of having an increased percentage of having a further delivery in 5 years' time. So here it's 84% compared to 70, here it's 68% compared to 52. So the work was sort of suggesting what our hypothesis was true. And in this last example, you can see the sort of proportion by mode of conception. I'm just going to move, sorry. I'm realizing I can't quite see the slides because of, yeah, okay. So we look at the proportion having ART for their second conception by whether the first child survives and by the mode of conception for the first child. Okay, we can see that if the child survives and the first conception was natural conception, 0.43% will have ART, compared with if there was death, 1.8%. And similarly, even when the first one is ART, you get this increase. So this shows nicely the carryover, and you can see that if we assume that the natural conception in the first pregnancy gives some indication that these women were not infertile, or certainly haven't been at the first one, they weren't using ART, and yet in the second pregnancy, when they've had a perinatal death, it's much more likely that they then go on to have ART. And, you know, clinically, there's a sort of a group of things that are related in terms of fertility and pregnancy outcome, which include miscarriage, stillbirth, preterm birth, and infertility. And it's about the ability to get pregnant and hold on to the pregnancy. And so we think what's happening here is sort of clinicians suggesting that ART may be beneficial, and is a more controlled way, I guess, than natural conception. So I won't go over this in detail. In terms of the last part of our notion that we would try to do some analyses where we could actually formally model these different parts of carryover and selective fertility and see if we can get closer results between the population conventional analysis and within SIBSHIP analysis. And that's presented in the paper. But the bottom line was, even with the sample size we had, it was too imprecise to give a robust answer. And the paper is, the pre-proof is available on the IJE website, and hopefully the proofed version will be there shortly as well. So I'm going to shift gear a bit for the last part of the talk and think about future cardiovascular health, and again, sort of some of the same issues that we've talked about. This I'm going to look at with record linkage and what we call the ART Health Birth Cohort Collaboration. And for both of these, the analyses are done based on people who have had a child. So it's not, we don't know outcomes for couples who used ART and didn't go on to conceive and have a live born child. So the first analyses are again from ConArtis. This is not work that I was involved with. A really nice study, again with the large sample sizes. And what you can see here is that in unadjusted analysis, there's some evidence for increased risk with those conceived by ART for cardiovascular diseases, type 2 diabetes, and obesity. When you adjust for observed confounders, and nicely they adjusted here for the parental cardiovascular disease, diabetes, and obesity before the offspring birth, which is really nice. And you see really for particularly cardiovascular disease and to a certain extent the type 2 diabetes, attenuation of the unadjusted results, less so for obesity. And then here are some analyses with another PhD student, Martina, from the UK, where we did some record linkage in the UK. Who knew that we could link records in the UK? We can, and we are increasingly more. So this was data on couples who'd had ART from a registry held by the Human Fertilization and Embryology Authority. And by law, any kind of infertility treatment has to provide a minimal set of data to that registry at the time of providing the treatment. So there's very good information on the treatment. This is an analysis. The paper was published quite recently, but the analysis is done on kind of old years. And the reason for that was in 2019, the HFEA changed the requirement for having individual participant consent to use their registry data. Prior to that, they didn't. And so for these analyses, we went just up to treatments going to 2009. We're working with the HFEA and the ethics regulators and couples undergoing treatment to do this with further follow-up with a larger sample. But what we looked at here was just a kind of simple outcome comparing to siblings and to the general population of any hospital admission was the key outcome, and then we looked by type of hospital admission. There was little evidence in either the conventional or the sibling analysis of a marked increase in population in hospital admissions, but some, and notably there were sort of increases for some neoplasms, respiratory diseases, digestive, musculoskeletal, but the opposite or sort of no associations for the kind of road traffic accident type outcomes, which behaves a little bit like a negative control outcome. In the sibling analysis, the results really attenuated. And if I focus, sorry, it's not a very good graph, but if I focus on the cardiovascular system outcomes, we actually didn't see any real evidence of an increased risk in either the comparison with the general population or the comparison with siblings. And just putting both of those results together, so the top three, the Kanatis results, where it's conventional multivariable regression with adjustment, including these nice data on the parents' risk, and the UK record linkage, where we look at the conventional multivariable regression and compare that to the within sibling analysis, it's reassuring in the sense that it doesn't suggest strongly an increased risk of cardiovascular, cardiometabolic diseases, possibly obesity in adults who have been conceived by ART. On the other hand, these are relatively young adults, and we may see something different with further follow-up. So lastly, I want to talk about this ART health collaboration. Currently, we have 26 birth cohorts from around the country. And the idea here is that if we take birth cohorts as opposed to the record linkage or the ART clinical cohorts, where the birth cohorts have recorded, or it can be captured from linkage, the mode of conception, we then have sort of a study where nobody has been selected on the basis of being conceived by ART, and everything from birth onwards is exactly the same. The follow-up, the confounders that are measured and so on is exactly the same between those two comparison groups. So our idea was that we couldn't do this with a single birth cohort, but if we could bring cohorts together, we may have some sufficient power to be able to add this to the mix of different types of study design. And if we had sufficient power to look over different ages, we could also look about whether we see patterns of changing outcomes with age. So this work has been led by a postdoc who works with me, and what he started with was looking at body mass index and other measures of adiposity. And what we see here, if anything, this goes from under 3 months up to over 18 years. We see that in infancy and early childhood, the ART conceived infants are actually lower body mass index. It sort of shifts towards the null, and there's a sort of very imprecise, because we've got smaller numbers, suggestion of an increased BMI in the kind of late adolescence, early adult outcomes. But again, largely reassuring. For the additional outcomes of waist circumference, body fat, fat mass index, there wasn't enough data to split by the same categories of age. But again, looking at, this is under 5 years, 5 to 10, 10 to 18, and over 18. And it's broadly a similar pattern. So waist circumference lower until we get to the older age. Body fat percent a bit more mixed, but generally lower until we get to the older age, and fat mass index lower until we get to the older age. Similar collaboration, but this time looking at cardiometabolic traits of blood pressure, lipids, glucose, and insulin. And again, broadly reassuring. Here, we didn't have data to split it by age group. So this is all of the ages combined, which is not ideal, but possibly lower systolic and diastolic blood pressure and no effect on heart rate, higher levels of triglycerides, HDLC, LDLC, total cholesterol, HDLC, slightly lower triglycerides, but nothing marked, and possibly reduced insulin, but that doesn't quite fit with the very null fasting glucose and HbA1c. And here we definitely need more data, larger sample size. We did have 3 cohorts that have repeat measures, and so what Hamed did was model the change over time, and we get a slightly different pattern now. This suggests that, again, going into adulthood, systolic and diastolic blood pressure change from being lower at younger ages to potentially higher at older ages. And we see sort of mixed patterns But here, the triglycerides start to look as if they're increasing with older age as well. So again, summarising those findings, these are observational data and the earlier sibling analyses. Mostly, I think these are reassuring, but it's largely relying on observational evidence, and it would be nice to be able to go forward with larger numbers and longer follow-up. So future plans, I've mentioned the UK record linkage, which in order to get more data and linkage to outcomes at older ages, we're going through the ethics process now, but we don't plan to do the linkage and get the data until the end of 2024 and look at it beyond then. We're interested in exploring molecular mechanisms in the different studies. There's this, within the collaboration now, we're just developing a pro form or a short A4 where people can put forward what outcomes they would want to look at. And in terms of what's coming next, Ahmed's very interested in looking at the relationship of ART conception with the offspring's reproductive health, and there's another group that are interested in mental health. And in terms of the methods, I'm keen to do more in trying to unpick the sibling analyses, thinking about appropriate negative control outcomes. Could we do Mendelian randomization here? My inclination is no, but some of my colleagues are doing a GWAS of ART use. And some of you may be aware that there are genome-wide association studies of statins and blood pressure and other treatments. I think it's an evolving field, and at the moment I'm at that stage where I'm trying to think more about it and just understand what these, when we do genetic predictors of treatments, what do we need to do in terms of the Mendelian randomization that takes account of the fact that the treatment is clearly for a condition. Just to finish, in terms of the cohort collaboration, Ahmed's the real driving force between this. These are the cohorts that are currently involved. If any of you have a cohort that you think might contribute to this, we'd really love to hear from you, so get in touch with Ahmed or me. And I will finish there by saying thank you. Many people involved in this work, but key people who have been the PhD students or postdocs who've really driven this: Ahmed, Kirsty, Martina, Singer, Maria Hazel, John have been some of the more senior team who've been invaluable. And thank you very much for listening.